Hello, everybody. This is one of the recordings to make up for lost time on that ice day on Monday. This is the conclusion to chapter one. So let's go to a screen share and we'll go through the slides. Tons of interesting information here. Let's see. And we're almost there. Can we make this full screen? I think it is. So we left off our discussion in the first chapter is why we need all these anatomical parts and physiological processes, the topic of AMP1 and 2. That's because we need to maintain homeostasis, this ideal living condition in the body, and different organ systems carry out different tasks. So to control homeostasis, I think in lecture I compared that to the working of a thermostat. And just like in our homes, in our bodies, homeostasis is continually disrupted, sometimes by external factors, heat, cold, lack of oxygen, and sometimes internal stimulus, exercise, um, eating a large meal, starvation, and includes psychological stresses as well. So disruptions are usually mild, and our body can respond to it, can restore homeostasis through the work of those organ systems. And if homeostasis is not maintained, disease, disorder is the result. And if it is very serious, we may perish. So how is this homeostasis maintained? So let's put a face on this mechanism, and we call these feedback systems. If the response by our body is reversing the original stimulus challenge, this is a negative feedback system. Most feedback systems in our body is of that type, and the thermostat in your home is also a negative feedback system. We have very rarely an example of a positive feedback system when our body's response enhances the original stimulus. We'll talk about an example here, very rare. So how is this feedback system fits into these organ systems? So we're going to take a look at that. In general, we have to find an organ system, cellular structure that monitors a controlled condition. We call this a receptor. There is a control center. If you go down this series of arrows, we need to detect the change in the environment. We have to have a control center to make a decision based on what these receptors are telling the control center. And then the control center says, okay, we'll need to restore homeostasis. Let's instruct organs, effectors to make that change. The best way to illustrate this is look at least a few examples. So here is maintenance of blood pressure. The challenge here is an increase in blood pressure. This could happen for many particular reasons. We have receptors that measure blood pressure. These are receptors called baroreceptors, but located in some blood vessels, and they detect the stretch of the blood vessels. The higher the pressure, the more the stretch. If these receptors detect a huge stretching of the blood vessels, they send nerve impulses, they inform the brain, and the brain is going to do two things. We need to lower the blood pressure back into the normal environment or normal measure. So our heart is going to pump slower, decrease in heart rate, and blood vessels will be dilated, increase in diameter. Both of those actions by the effector organs lower blood pressure, and hopefully all those changes in the body, in the effectors, will normalize blood pressure. So this is an example of negative feedback. We had an increasing blood pressure and we lowered it. We reversed this change back to normal. Positive feedback. Now this is an example of that very rare situation. We have altogether 2 of these listed in our textbook. In chapter 28 in AMP2, we'll see a little bit of this childbirth in connection with the reproductive system. But here, let's take a look at this. So here's a situation, end of 3rd trimester. As you can see, here's a fully formed fetus. And occasional contraction of the uterus is going to push the head of the fetus into the small structure here called cervix. And the stretch receptors in the cervix will send nerve impulses to the brain. The control center will respond by making a hormone called oxytocin. And oxytocin will instruct the effectors, the smooth muscles of the uterus, to contract. So instead of lowering the initial challenge, the stretching of the cervix, more contractions will push the fetal head into the cervix even more forcefully, so the stretching will increase. So this is a positive feedback. Now, this cycle of events continue sometimes for several hours. Labor is the name of this process where the stretching of the cervix makes the brain to release a hormone, and that hormone is going to amplify the contraction of the uterus. And this positive feedback will accelerate until we have the delivery, and the birth of the baby will bring this process to an end. So in the positive feedback, we do not reverse the initial challenge. Quite opposite, we are enhancing it. The end result is a biological outcome, in this case, the birth of the child. Again, positive feedback mechanisms, extremely rare. To participate in these feedback mechanisms, we need individual molecules to participate. Think about oxytocin is a protein molecule. We need cells to participate in these responses. Think about the baroreceptors or neurons, cells that monitor the condition. Tissues participate too. Think about the smooth muscle tissue of the uterus in our example of the positive feedback. Organs participate. Think about the heart, blood vessels that participated in that negative feedback, or the brain as a control center. And organ systems also play a role in the maintenance of positive and negative feedback, maintenance of homeostasis. So because these levels of organization are involved, we are going to start looking at these in chapter 2. Let's take a look at least a little bit how chemicals are involved in maintaining homeostasis. And then we move on to studying the cells a little bit. In chapter 3, chapter 4 will be our tissues. So your textbook is following this level of organization for the first few chapters. Here in chapter 1, in addition to talk about homeostasis, there is a segment on basic anatomical terminology. We're going to see a short segment on very basic body structures, such as cavities in the body. And we also have a closing segment on medical imaging. The basic anatomical terminology, anatomical position, body regions, planes and sections will be covered in good detail in the lab. And so I'm going to go through this relatively quickly. So basic anatomical terminology, language of anatomy is needed. So any part of the world can take a medical instruction, description, and apply it for their own use. And anatomists found it wise to fix the body in an anatomical position for any kind of clinical discussion. So here's a body in anatomical position, standing up straight, looking forward, and palms are pointing forward. You can see the thumb sticking out to the side. These are hallmarks of the anatomical position. On this anatomical position, the body parts, organ systems are described along the major directional terms. So if you want to describe something which is in the forward portion of the body, we use the word anterior. If we want to describe a body part that is in the backside of the body, we call that posterior. And we can say that, for example, take a look at this. The sternum is anterior to the heart, because the sternum is more towards the front of the body, while the heart is behind it. We have up and down terminology in this anatomical GPS. And so superior, inferior means that superior is above, inferior is below, along the long axis of the body. And we can say, for example, that the stomach right here is inferior to the lungs, meaning it is situated below along the long body axis. On the limbs, we use above and below by two different Latin terms, proximal and distal. On the limbs, upper and lower limbs, proximal means above, distal means below. So we can say that the arm right here is proximal to the forearm, because the arm is above of the forearm on the limb. We also have this lateral medial directional term. This is measured out from the midline of the body. And we can say, using this medial lateral terms, is that the lungs are lateral to the heart. The lungs are further away from midline than the heart is. These terms are used extensively in your textbook and lab manual, and we will be practicing these in the lab. It is also important to talk about anatomical planes. These are standard cutting surfaces that cut the body according to an agreed, planned surface. This is again identical in all parts of the world. So medical descriptions, imaging, can be described using these planes. So let's demonstrate this. So sagittal plane is dividing the body into left and right portions. You see the mid-sagittal plane here, dividing our body into equal left and right portion. The para-sagittal plane is dividing the body into unequal left and right portions. We have the frontal or coronal plane. You can see the representation right here. It divides the body into anterior and posterior portions. And we have a transverse plane that divides the body into superior and inferior portions. We also have a rarely used oblique plane. We may say a little more about this in the lab. So these are parts of anatomical language. Again, these planes are used a great deal in your textbook and lab manual to illustrate internal structures of the body. Here's what you see when you apply this to the brain. So the advantage of using these planes is that when the brain is cut along the mid-sagittal plane, certain set of structures are visible. They can be described. If somebody cuts the brain along the frontal plane, a different set of structures come to view. Some of them were not visible in the mid-sagittal plane. And so there's an organized, planned out exploration of any body part. Here the brain is our example. After the anatomical language, let's talk about some general body parts. These are not necessarily maintaining homeostasis, although we'll see that later on, especially in MP2, we will explore some functions to them. But consider these body cavities are part of the body plan. How is the body built? And we are not built as a solid mass of cells, but we have empty spaces, body cavities that contain organs. So here are the body cavities holding our nervous system organs. We have cranial cavity that holds the brain. We have the vertebral canal that contains the spinal cord. With every cavity, there are membranes surrounding the organ or lining the cavity. And so the brain and the spinal cord surrounded by the membranes called meninges. For visceral organs, we do have the thoracic cavity. This is situated superior to the diaphragm. And we have the organs, lung, heart here. And then we have the abdominal cavity. So let's take a look at, oops, sorry. The thoracic cavity is presenting a little more complex picture. We do have the pleural cavity. This purple area here is the pleural cavity. It is bordered by two membranes, the parietal pleura and the visceral pleura. And a pleural fluid in purple here is filling the cavity. The pleural cavity surrounds the lungs. There's a very similar situation with the heart. There's a pericardial cavity made up by the parietal and visceral pericardium. This brownish liquid here is the pericardial fluid, and this pericardium surrounds, or pericardial space surrounds the heart itself. There is an additional space in the thoracic cavity called mediastinum. Organs here do not have a covering membrane. organs that are present here are esophagus, trachea, some large blood vessels. And the last segment here in this lecture is medical imaging. So we just talked about body cavities. If you want to look into somebody's body cavity, you have to cut the body open, right? You either cut open the cranial cavity, thoracic cavity, abdominal cavity. And here comes the neat aspect of medical imaging, looking into the body without cutting it open. Huge, right? So this started over 100 years ago with X-rays. This is called traditional conventional radiography. X-rays penetrate into the body, and we are detecting these X-rays either bouncing back or passing through the body. And we obtain this image. This is tremendously useful to image bones, although radiography has been modified to create CT, tomography, X-rays, radiography is used in mammography, and there are other modifications. So structures other than bones can be imaged given the right modification of this radiography. With ultrasound imaging, it's high-pitched, high-frequency sound waves penetrate into the body, and we detect them using a detector. The resulting image, which we call a sonogram, is a very detailed description of internal organs. For fetal ultrasound, not only the face of the fetus can be imaged that you see here, functioning of the heart, construction of kidneys, internal structures, skeletal structures are also studied. Abdominal organs such as ovaries, intestines, other structures can be modified by ultrasound. Even blood flow through blood vessels can be done using Doppler ultrasound, a modified version of the ultrasound technique. MRI, this has also been modified for several specific use. In this case, radio waves penetrate through the body. And if the body is in a high energy magnetic field, these bouncing radio waves create a signal that using a computer, we can turn into an image. And soft tissues are especially imaged well. You see here the brain, but tendons, ligaments, and many, many soft tissues internal to the body can be used to image. And the last one is PET or positron emission tomography. This is the imaging technology where a tracer has to be injected into the body, and the detectors outside the body are picking up the energy, the gamma rays released from the injected tracer. So what you see here is a brain, and you can see the tracer activity either high with the red or low with the green. But then here's a portion of the brain where the tracer never gets into. Either some trauma, maybe stroke, prevented the tracer to move into this body part. And so imaging can be done using positron emission tomography. So a tracer is emitting the rays that we can detect outside the body. And this is it for this lecture. I'm going to stop sharing here. I'll see you guys in the lab, and we'll talk about many of these topics. I will attach this before you come to the lab. So if you have any questions, please let me know. And have a great rest of the day.